Hi everybody. In the previous video we have seen the basic aspects of the gene regulation in case of prokaryotes. We have seen the structure of the gene, then what is an operon system, how you can classify the operon system, all those things. Today we are going to concentrate a very important aspects in molecular biology, that is nothing but lac operon system. So as all of us knows, that is the basic requirement for the growth of an organism is nothing but the carbon source, mineral source, nitrogen source, all those things. So the carbon plays a very important role, and we know the basic concept that if glucose and if you want to go in detail, that is the carbon source can be either a monosaccharide, it can be a disaccharide, it can be a polysaccharide, but if a monosaccharide or a simple carbon source is available, always the organism will go for what? The simple carbon source like your glucose. If that glucose is not available, then only it will switch on to the complex carbon source, depends upon the complexity. So just imagine a media which contains both glucose as well as lactose, and you are inoculating an organism into that, it will show a dioxic growth pattern over there. What is a dioxic growth pattern? See, what is the simple source of sugar which is present in the media? Nothing but the glucose. So the organism will adjust for that new environment, that is it will adjust for the glucose environment, it will create a lag phase for glucose, then it will start utilizing the glucose exponentially and it will reach into the log phase of the glucose. So once the glucose is exhausted from the media, then the organism doesn't have any other option, it has to go for lactose. So to utilize lactose, it requires so many basic machineries. For that it will adjust for the lactose environment, so you will get again a phase called lactose lag phase. Then it will utilize the lactose and reach into the exponential phase of the lactose and reach into the stationary phase once the lactose is exhausted, and finally into the death phase. So this is what this is called the dioxic growth pattern of a bacteria. So if you see the dioxic growth pattern, you can see, you just imagine, I will recall once again, you will be having glucose and lactose. So the first phase is nothing but the lag phase for the glucose, next is the exponential phase for the glucose, next lag phase for the lactose, then comes the log phase for the lactose. So this thing is this particular growth pattern is called what? Dioxic growth pattern. Or simple way you can tell that the tendency of the organism to utilize the simple sugar first, then switch on to the complex sugar is called dioxic growth. It is the tendency of the organism to utilize simple sugar first, then switch on to the complex sugar is called what? The dioxic growth. So the conclusion in this particular thing is that is if glucose is present, then obviously it will go for glucose, and if the glucose is not there, it has to go for lactose. So to go for the lactose, the E. coli or the prokaryotic organisms will be having a basic mechanism called lac operon system, which has been described in the 1940 to 1950s by Jacob and Monod, and they have been awarded with Nobel Prize in the year 1965 for the wonderful work in molecular biology. So if you see the lactose operon system, if you see, we have seen that the prokaryotic genome is it is not a monocystonic mRNA, it is a polycystonic mRNA. So we have discussed yesterday what is a polycystonic mRNA. It contains many sequences, many coding sequences, unlike your eukaryotic mRNA is monocystonic. So if you see the genome of the lac operon system, you will find the various regions over there. So let us discuss what are all the various regions in case of lac operon system. The first one is a lac I. Okay, in the downstream, yesterday we discussed what is the downstream and what is upstream. In the downstream you can find a region called lac I. Lac, which indicates the lactose, I indicates it is an inhibitor, it produces an inhibitor protein, that is called what? Lac I. Then you will be having a region in the upstream only, that is promoter operator region. We have discussed what is the function of the promoter operator region yesterday itself. That is, the promoter is the region where in which what happens, the RNA polymerase will come and bind and it will transcribe your entire genomic sequence. Next comes operator region, whatever the repressor protein or inhibitor protein is present, that will come and bind into the operator region, which will inhibit the action of RNA polymerase towards the promoter and stops the transcription. Along with this particular regulatory region, there is one more region called CAP region. CAP is nothing but catabolite activator protein region. We will see what is the function of catabolite activator protein region. Then comes three structural genes. The lac operon contains three structural genes, lac Z, lac Y and lac A. So each of the structural gene is responsible for the production of a particular protein. For example, LAC-Z is having the capacity to produce, or it is responsible for the production of an enzyme called beta-galactosidase. So the function of beta-galactosidase is very simple, that is, it will convert the lactose into glucose and galactose units by breaking down the glycosidic bond, beta-glycosidic bond, which is present in the the glucose and galactose. The next sequence is nothing but Y. The Y is responsible for the production of beta-galactosidase permease. It is a membrane-bound protein, it is a cytoplasmic membrane-bound protein. So it is bounded with the membrane, and which is responsible for the transportation of the lactose from the media inside the cytoplasm. So it acts as a permease enzyme, that's why it is called beta-galactosidase permease. Right? The next enzyme is, the next sequence is nothing but A. This region is responsible for the production of an enzyme called trans-acetylase. So this trans-acetylase, it will transport acetyl-CoA into galactosides, which will increase the efficiency of it. The function is not completely understood, but it is proposed that it increases the capacity or the functioning capacity of the enzyme, that is, it transports acetyl-CoA into galactoside. So these are all the structural genes which are present in case of lac operon system. So LAC-Z, beta-galactosidase, conversion of lactose into glucose and galactose. Y, membrane bound protein, which is responsible for the transportation of the lactose from the media inside the cytoplasm, and an additional component, that is galactoside, the trans-acetylase enzyme, which will transfer acetyl-CoA into galactoside. Let us see the different scenario, that is what will happen in the presence of lactose and in the absence of lactose. In the absence of lactose, there is no necessity for the production of enzymes which are responsible for the metabolization of what? Lactose. So what will happen is, LAC-I is continuously producing genomic sequence which produces the inhibitor protein. So this particular inhibitor protein will go and bind into the operator region in the operon system. So once it binds into the operator region, RNA polymerase cannot come and bind into the promoter region, and transcription cannot happen. So there will not be any production of all the three enzymes whatever we have mentioned, that is beta-galactosidase or permease or trans-acetylase, nothing will be produced. There is no processing of your lactose happens over there. But whenever we tell that the operon system is shut down, it does not mean that it is 100% shut down. It is minimally expressed actually. So when a lactose is observed in the media, the minimally expressed lactose permease enzyme will transport this lactose from outside environment into inside environment. A minimum quantity of lactose will be transported from the media into the cytoplasm. So one more thing this beta-galactosidase will do, permease already transported the lactose inside the cytoplasm, and the minimally expressed beta-galactosidase, what it will do is, with the association of lactose permease, it will convert a small amount of lactose into allolactose. It is an isomer. Okay, it will convert a small amount of lactose into allolactose. So once the allolactose is present, this allolactose will go and interact with the inhibitor protein which has been already bound into the operator region and change the conformation of this inhibitor protein. So once the conformation is changed of the inhibitor protein cannot stay back over there. It will detach from the operator system. So the promoter is free there. So RNA polymerase is free to come and attach into the promoter region, and it can go for transcription, synthesis of all the necessary enzymes, and more amount of beta-galactose, permease enzyme produces, more amount of lactose is transported into the media, more amount of beta-galactosidase is produced, so more amount of conversion of lactose into glucose and galactose. So the lactose is converted into glucose, then it can synthesize energy, that is ATP can be synthesized. This is in presence of lactose. If you see in the absence of lactose, there is no need of synthesis of all this enzyme. So the inhibitor protein from the LAC-I region, it will continuously synthesize. It will go and bind into the operator region. So once it goes and binds into the operator region, there is no chance of RNA polymerase comes and bind into promoter region, and there is no transcription process happens over there. So this is how the regulation happens. So in presence of lactose, the operon is switched on. Yesterday we have discussed already that it is an example of inducible operon in this case. I told that the lac operon is an example for both positive as well as negative regulation. So you just imagine the glucose is present. We have discussed that scenario previously. So if glucose is present, how the lac operon behaves? So we know that if glucose is present, obviously it will go for what? The glucose utilization, and it will avoid what? Lactose. So how this mechanism works out is, in the presence of glucose, the concentration of cyclic adenosine monophosphate level in the cytoplasm will be very less. Means glucose is high, CAMP level will be low. It is vice versa also. That is, the glucose is less, CAMP level will be high. So what is the importance of the CAMP in the gene regulation is, if the CAMP is present, it can bind into a protein, a cytoplasmic protein called the catabolite activator protein, and it produces a complex called CAP-CAMP complex. What is CAP? It is nothing but catabolite activator protein which combined with CAMP. What is the CAMP? It is cyclic adenosine monophosphate. And we have discussed in the structure of lac operon, there is a region called cap region along with the promoter region. So it will go and bind into the cap region, which will go and bind the CAP-CAMP complex will go and bind into the cap region. So once this complex is bounded into the cap region, it increases the transcription ability. That means, so this complex is having the capacity to load more and more RNA polymerase into the promoter operator region. So once the promoter operator region is loaded with RNA polymerase, what will happen? Transcription happens, okay, and there will be a production of all the enzymes, and the lactose will be utilized. So this is general mechanism which I told you, that is, if glucose is very less, CAMP level is high. If CAMP is there, CAMP combines with CAP. This particular complex will go and bind into which region? Cap region of the operon. Once it is bounded into the cap region, what will happen? It will load more RNA polymerase and the transcription continues. So this is what the basic mechanism. So you might have understood what is the concept, even though I will just give you a scenario. In presence of glucose, what is the result? CAMP level is very less. So if the CAMP level is very less, what will happen? It will not bind into the cap, that is catabolite activator protein which is there in the cytoplasm. So if this complex is not produced, it will not go and bind into the cap region. Therefore, the transcription will not happen. It will not load the RNA polymerase. It will utilize glucose and it will happily multiply in the media. The second scenario, in the absence of the glucose, CAMP level is high. Okay, once the CAMP level is high, it will combine with what? Cap. So it produces a complex called the CAP-CAMP complex. It will go and bind into the promoter, it bind into the cap region. So once it binds into the cap region, what will happen? It will load RNA polymerase into the promoter operator region, and transcription continues, and it synthesizes all the necessary enzyme for the metabolism of the lactose. So this is how the regulation, the dual control of lac regulation happens. One is in presence of glucose, how it acts, and the absence of glucose, how it acts. So totally, if the glucose is present, it will go for glucose, then it goes for lactose. So this is how the lac operon system works out. To concise the entire topic, that is, you just think a scenario, that is, glucose is absent, lactose is present. If the glucose is absent and lactose is present, what will happen? The lactose will be transported inside by the permease, and some amount of the lactose is converted into allolactose. So already the inhibitor protein is present in the operator region. So it will go and bind into the protein, that is inhibitor protein, change the conformation, detach that protein from the operator region, and RNA polymerase will transcribe. That is in presence of lactose. In the absence of lactose, what exactly happens? That is, there is no need of synthesizing all this enzyme. Therefore, inhibitor protein remains in the operator region. RNA polymerase cannot come and bind. Therefore, the operon shut down. In presence of glucose and in the absence of lactose, what will happen? If the glucose is more, there will be more synthesis. And the CAMP level is very less. Therefore, it will not bind into cap protein. There will not be binding of cap protein in the cap region, right? So if it is not binding, there is no loading of RNA polymerase. It utilizes glucose and multiply. In the absence of glucose and in the presence of lactose, in the absence of glucose, CAMP level is very high, right? And CAMP will combine with the CAP. It will go and bind into the cap region, and it will load more RNA polymerase, and the transcription continues. This is how lactose metabolism is controlled in case of E. coli.